Hello guys, welcome back to my channel. Today we are going to solve some GATE BT 2022 MCQ and MSQ type of questions. So let's proceed. First question is, in animal cells, the endogenously produced miRNAs silence gene expression by. This is MCQ type of questions with 1 mark. First is base pairing with 3' untranslated region, that is 3' UTR of specific mRNAs. Next is blocking mRNA synthesis. Next, binding to the operator site. Last is base pairing with 3' region of specific rRNAs. So miRNA, that is microRNA, it is single-stranded RNA with 22 nucleotide. It actually silences mRNA by binding with its complementary region. So the blocking mRNA synthesis will not be correct. Next is binding to the operator site. miRNA binds with mRNA in mainly untranslated region, not the operator site. So this will be again wrong. Base pairing with 3' region of specific rRNA. No, it is, if it is mRNA, then it can be true. So the option D again wrong. Base pairing with 3' UTR region of mRNA. This is right. miRNA actually binds with 3' UTR of targeted mRNA to silence its expression. So the option A will be right answer for this question. Next is match the microbial product in column 1 with its respective application in column 2. This is again MCQ type of question with 1 mark. Methane, next is glycolipids, third one is polyhydroxyalkanate. Methane is the simple alkane which is a gaseous in nature and it is used as fuel. So it will be biofuel in nature. So P3. Next is glycolipid. Glycolipid means lipid attached with sugar. Lipid attached with sugar. It is found in mainly plasma membrane. It also acts as biosurfactant. So Q1. Next is polyhydroxyalkanoate. It is produced by sometimes bacteria through the fermentation process. And it is also used as plastic which is biodegradable. So it will be bioplastic. So R2. So P3, Q1, R2. So this is option is D. So option D will be right answer for this question. Among individuals in a human population, minor variations exist in nucleotide sequences of chromosomes. These variations can lead to gain or loss of sites for specific restriction enzymes. Which of the following technique is used to identify such variation? This question is MCQ type with 1 mark. Genetic marker actually distinguish or identify variations. Variations identified by genetic marker. Genetic marker are of 2 types. These are dominant and codominant in nature. Dominant genetic markers identify homozygous type of conditions and codominant marker identify homozygous as well as heterozygous type of conditions. Homozygous and heterozygous means what? Homozygous means only from one parent, while heterozygous means from both parents. Dominant markers are single allelic and multi-locus in nature, while codominant markers are multi-allelic but single locus nature. The example of dominant genetic markers are AFLP, next RAPD, next ISSR, and codominant markers examples are SNP, RFLP, SSR, STR, VNTR, sometimes AFLP as well. It is mainly dominant in nature, but sometimes AFLP may show codominant type of genetic marker significance as well. So according to this table, polymerase dependent fragment insertion will be wrong. Real-time polymerase chain reaction, this is again wrong. Restriction fragment length polymorphism, that is RFLP, which is included in codominant genetic marker. And last one is reverse transcriptase polymerase chain reaction, that is RT-PCR. It is again wrong. Real-time polymerase chain reaction is quantitative PCR, that is qPCR. So the right answer will be option C, that is restriction fragment length polymorphism, that is RFLP. So this is for today's session. Hope this will be helpful for you. Thank you for watching and keep learning.